Okay, hello everyone, and welcome to the Neurons Matter seminar. We'll still have a few more people joining us, I'm sure, over the next couple of minutes. Today's seminar is going to be given by two different presenters. They'll each take about half an hour, and I'll introduce them in a second. This seminar is a little bit unique as compared to those that we've had in the past because it's associated with the summer school that is going to be coming up in the summer of 2025 that we're planning for June 22nd through the 28th, 2025 in Linkoping, Sweden. And as part of that, we plan to put out a call for proposals for collaborative projects for those who are interested in performing joint experiments in microneurography at the summer school. And the idea is that we could collect preliminary data. Those investigators could potentially collect preliminary data that could be used for subsequent efforts. And so we will have a process that we'll be laying out that'll involve a webinar and a pre-application and some matching of a project to investigators. And those that are ultimately selected, we're able to provide mini sabbatical funding on the order of up to $7,500 US funds to U.S. institutions. So details on this will be forthcoming, but we wanted to couple that announcement with these two particular talks because both of these two investigators are in Linkoping and would be associated with this particular effort. Okay, so I'll jump into our first presenter. And Sarah, if you want to bring up your slides, that's fine. Sarah McIntyre established the Lab for Haptics, Touch, and Mechanosensation Research at Linkoping University in 2021. It's a multidisciplinary team incorporating expertise from neuroscience, psychology, and engineering. Dr. McIntyre's research focuses on understanding human touch sensation using a variety of techniques to address questions from a whole system perspective. This includes measuring skin mechanics, peripheral neural coding, and human perceptual judgments. A key focus is on characterizing and understanding the full range of touch inputs that humans experience in the world and how the nervous system can process this huge variety of signals to produce functional outcomes. Thank you very much, Sarah, for presenting today, and please go ahead and take it away.
All right, thanks, Greg, and thanks to the NeuronS Matter Network for having me. As Greg mentioned, this is a two-part presentation. So I'll talk about touch and with a particular focus on affective touch and how we can use microneurography to investigate questions about affective touch. And I think the original title said we would also include proprioception, but just in the interest of time, Saad and I decided just to focus on touch and nociception. So to start with, I want to just mention briefly, so affective touch, for those of you who aren't familiar with this term, this refers to sort of emotional touch, or affective refers to something which has an emotional component. So that can include like social touch or pleasant touch, which isn't social. It can even include unpleasant touch, but usually when people talk about affective touch, it's about pleasant touch. And if we're talking about the nervous system, one question we can ask is, where does affective touch begin? And when we're doing our microneurography recordings from the peripheral nervous system, we are recording from these first-order neurons. They don't have any direct influence from central activity, central neurons. So in a sense, they're like, you would think, okay, maybe it's a little bit of a tricky to think how is affective touch going to be embedded in these sort of first-order neurons, which are just responding to mechanical inputs from the environment. But where surely all the emotion is happening in the brain. But if we think about, for example, if you want to create a scale to measure humans, or you want to make the same to weigh humans, or if you want to make another scale to weigh elephants, you're going to have different properties for those scales. Like they're going to have different range of sensitivities to different weights. So the idea is that if you look at these sensors in the first order neurons in the periphery, can we see that they're like tuning in their response properties that is relevant for processing affective touch and social touch? And when it comes to social touch, the kinds of mechanical inputs that we can consider, whether if the neurons might be responsive to, is you could think about the force or the pressure, how the skin deforms, what is the contact area, how many sort of points of contact do you have. You can also look at components of motion, the direction, speed, distance. You can also think about things like softness of the material that's interacting with the skin, and conformation, like if with a hug or a squeeze, there's sort of conformation around the surface of the skin. So these are just some of the things we can think of. And so if you want to look at how the mechanical inputs present in social touch are influencing the nervous system, then you can look at the primary afferent responses, these first order neurons that is responding to these mechanical inputs, and see how their response properties are affected in relation to affective and social functions. And so just a little introduction about microneurography here. We have, this is how the method works. Basically, is we in the peripheral nerve, go zoomed in peripheral nerve here, which actually has a bunch of little bundles inside, fascicles, and we have an electrode that we insert into the nerve and try to get it very close to a single neuron axon and record just from a single neuron. And you can see on the right there, this is an ultrasound. We use this ultrasound guidance system to get closer to the nerve. But basically, once it's in the nerve, we're just having to make small movements, and it's like fishing. We're kind of running blind and just basing on the signal that we get. We try to make sure that we're recording from a single neuron. And this is done in healthy volunteers that come into the lab, and we'll just insert the electrode usually through the arm. And so it's very small electrode, very thin, and it's thinner than acupuncture needles. And this has been safely used for many decades, mostly in Sweden. Excuse me. And so the other thing to consider is that not all of the neurons that we record from are the same. So, and there are a lot of different ways of classifying them, a few different schemes. So one is if we can look at the axon myelination. So if the axon is myelinated, then you could see this in anatomical studies by looking at the axon, or you can also detect this in microneurography because the thickly myelinated axons are conducting the signal much faster. And with the unmyelinated axons, there's a delay. And we can also look at the stimulus response profile. So these two are the two classification schemes that we can directly use with microneurography. So the stimulus response profile simply look at how, as you change the stimulus, how does the activity of the neuron change or differ from each other. And there are other methods. You can look at receptor morphology, or you could look at the molecular profile, but these methods are not directly available with microneurography, but we try to compare results across studies to find correspondence between them. Okay, so just a little bit more about afferent classification. Within microneurography, you can see that in the top left here, if you see a trace, this is an indentation trace of a stimulus. And as the pressure is applied to the receptive field of this neuron, it responds. But then while it's being held, it goes silent. And when it is removed again, then the neuron responds again. This is an example of a sort of fast adapting, is also called rapidly adapting in some papers. And this is contrasted with, for example, slowly adapting neurons, which when they continue to fire the whole time that this indentation is held. And you can see also that these two, they basically respond straight away. As soon as the indentation occurs, there's not really any delay or very little. And this is, so these are thickly myelinated, the A beta fibers to indicate the myelination. And this is in contrast with C tactile fibers, which are unmyelinated, and you can see this delay here from the response to the C fiber. And they're also called CLTMR, C low threshold mechanoreceptors. And just this, yeah, a bunch of different labels at different times and for different reasons. So it's good to be aware of this. And so there's just some examples of how to classify. I won't go into all the details of all the classes that we have, but here are the classes of primary afferents that we observe in the hairy skin that are touch sensitive. And this is sort of over many decades of research, the stimuli that people have observed, they seem to be sort of optimally, or they seem to prefer the stimulus. They respond very well to these stimuli. That doesn't mean that's the only thing that they respond to. And I'll just quickly mention, for the sake of the proprioceptors, these muscle spindles. So these are, yeah, generally thought of as proprioceptive neurons, but they do respond to touch, and I won't talk about them very much, but just a nice mention. So if we go back to affective touch, a very influential study I want to mention is this one, which really kind of, I think, solidified this like CT affective touch hypothesis, which is, excuse me, that there's one class of primary afferents, C tactile afferents, have a special role in signaling affective touch. And the mechanical stimuli that they focused on in this study was brush stroking velocity. So they use this kind of device to deliver well-controlled brushing with a nice, gentle, soft brush. And they had a very wide range of velocities. And what they found was really a very different pattern in the CT firing responses to this brushing stimulus compared to the other afferent types, which were all A beta. So these are thickly myelinated afferents, and this is the unmyelinated. And what they found is this inverted U shape response to the brushing velocity, so that as the optimal response, the preferred response, is this like around 1 to 10 centimeters per second. And whereas all of the other afferents, they just increase the firing frequency the faster you run the brush. And if you compare to psychophysics here, this is people's pleasantness ratings for this same stimuli. You can see that the preferred CT response corresponds to the preferred, to the most pleasantly rated range of velocities. So the sort of simple version of this is that, okay, so CT is affective touch and A beta is discriminative touch. This is a bit of an oversimplification. There have been a lot of studies that are inspired by this and related work, which are kind of digging into this relationship. But for the purpose of this talk, I just want to mention that simple version of the story, which is broadly what you will encounter in the literature. Okay, so in now I want to talk about a study that I did when I first came to Linkoping, and I was working with Håkan Olausson. And we were interested in, okay, not just this like pleasant brushing, but how do people actually use touch? What are all the different kinds of like mechanical inputs? What sort of strategies do people use to communicate when they're using touch in a social context? So we had an experiment where people, we brought people into the lab, pairs of people who already knew each other with an existing relationship that they were happy to touch each other. One of them was assigned the role of the receiver, one the role of the sender, and the sender was cued with a message, something like your partner is making him or herself upset, thinking about a situation that cannot be changed. Try to be calming through touch. And then this person, the sender, needed to just communicate that message only through touch, and there's a curtain so they can look at each other, and we asked them to be quiet. And we, for the purpose of our microneurography study, we did a few different studies with this, but the purpose of the microneurography study was to generate a lot of different touch expressions so that we could see what physical strategies people use. But we had a variety of different social touch messages that people tried to convey. And if we look, based on what people did, we came up with sort of standardized touch expressions for 6 of these messages. And we looked at the strategies that were used in the successful touch communication trials. And we sort of avoided the strategies when the receiver didn't successfully guess what the sender was trying to say. And we also considered things like in the selection of these stimuli, some practical issues for microneurography. So we didn't want to have anything that introduced too much movement of the arm, because this might dislodge the recording electrode. And then, yeah, it should be sort of just located, easily applied to the receptive field of the neuron when the person's arm is lying very still. So we didn't investigate other touch interactions like hugging or whole body interactions, because we wanted to be able to use these expressions in the lab. Okay, so when we did these nerve recordings, we got a number of different types here. So I mentioned the classes of primary afferents before, and you can see here these are the locations of their receptive field. So primarily on the back of the hand and around the wrist, and most of the muscle spindles that we recorded from were on the arm. And this is just to show you sort of the extent of the location that the neurons were responding to. And it's important to note that for these touch expressions, that all of these afferent classes responded to the stimuli. And here's an example of how it looks in the lab. So we have the nerve trace here. So that's a single SA2 afferent that's responding to this gratitude expression. And so the relatively raw signal that we get here of the nerve response, we sort of turn that into single spike events. So we log the time of the events and we get this spike train. And we can also look at the instantaneous firing frequency to see when the sort of peaks of biggest activity were occurring. And then, and for this project, we also teamed up with Greg Gerling and his group at University of Virginia. And Sean and Stephen were leading the analysis efforts. for this. So they trained a convolutional neural network, a classifier, to basically take as input the spike trains from these touch expressions and get as output, assign the label of what was meant to be expressed. So here, if you look at this confusion matrix, what this is showing is the true or intended touch expression message. And this is how the classifier has labeled it. And so if you get this nice solid blue on the diagonal, that means the classifier is mostly getting it correct. And the overall accuracy is around 80% for the SA2s. And we also have very good classification accuracy for HFAs and field units. Whereas the FA2 muscle spindles and CTs were not so good at discriminating these touch expressions, at least in our sample that we reported from. And just as a reminder, I'll show that, so this is from previous work, the sort of preferred stimuli for these afferent classes. So the hair follicle afferents are responding to hair deflection, and the SA2 afferents are responding to skin stretch and pressure. But our results are showing that the mechanical sensitivities that they have, it may be particularly suitable for discriminating different kinds of social touch inputs. And it's very interesting to note that the CT afferents, although they respond, they're not discriminating these different kinds of social touch expressions very well. And I should also point out that this is, we're recording from single neurons, but of course, all of these touches are activating many neurons simultaneously. This is a limitation of the recording technique, that we only record from one neuron at a time. Presumably the brain is actually using inputs from not just single neurons from, not just neurons from one class, but this analysis nevertheless tells us about how well these particular classes are tuned to the features present and useful for the discriminating social touch. And we also looked at the time scale of social touch expressions, and we can see that if you adjust the window length, and we did this like in different ways, so not always from the beginning, but so if you take the spike train and just take a subset of it, we can evaluate how much time is needed, or how much data, or how much of the spike train is needed for the classifier to be able to judge or discriminate the expressions based on that input. So we can see that the SA2 and HFAs and the fields, they're reaching this plateau here, a very good performance. I mean, it's there for the full window length, but it's still very good even around after like 3 to 5 seconds. And when you consider this in contrast with sort of discriminative tasks, like if I'm holding a glass of water and it starts to slip, I need to react very quickly in milliseconds. But for a social task, like trying to figure out why this person's touching me, what they mean by that, then this is kind of an appropriate time scale to be able to extract that information. So we can see that the nervous system is prioritizing this, the time scale is appropriate for the priorities according to urgency. Okay, so just a little summary of what I've covered so far. So we find that we do find peripheral tuning to social touch mechanics, particularly in hair follicle afferents, SA2s, and to some extent field afferent classes. And this is occurring at a sort of functional time scale. And this is done on a single unit level of analysis. So it's always good to keep in mind when you're trying to make these inferences back to perception and function, that the brain is certainly using not just single neuron inputs. And where does this leave us with the CT effective touch hypothesis. Okay, so the CT, so one detail of the effective touch hypothesis is that the CT, people have found that previously that the CTs are not very good at discriminating all sorts of stimuli, and that it may just be a signal that some affective or affiliative touch is occurring. And and the A beta afferents are for discrimination, but what we've shown is basically that it's not this like affective versus discrimination, but the A beta afferents are discriminating affective stimuli as well. So this is, I would say, just adding on something to this CT effective touch hypothesis, which is not really contradicting it at all. And so another limitation of the work I presented so far is that we don't know really which features of the social touch these afferents are responding to. And so some additional work I want to mention is our efforts also with Greg Gerling's group at University of Virginia is to measure contact characteristics. I won't be showing any data right now because we have data from behavioral studies published, but we have done some recordings measuring contact characteristics of human touch with microneurography, but we haven't finished analyzing that yet. So, but briefly, we can see that this, if we have a camera set up here, and in the behavioral study, we have a similar toucher receiver setup, and we can make a really nice sort of high density vector model of the forearm, which is sort of specific to the individual. And we also have a model of the toucher's hand, and we can, in 3D space, detect the collision of the boundaries of these two modeled objects and detect contact and get various contact characteristics like contact area, depth, and velocity in 3 dimensions. So this gives us a really nice estimate of actually the mechanical inputs that are present, and we can do this. We've recorded in real time with microneurography. So this is sort of something that hopefully you'll be seeing soon. So here's just an example of how this tracking system works. This is during a microneurography experiment. And you can see how the contact characteristics are changing over time. This system that I'm showing here, this was developed by Shan Zhu, and an earlier version was developed by Stephen Hauser. And so just to finish up, we have current focus areas. Also, don't have any data to show on this, but just show the capacity in the lab. One of the things we're looking at is functional skin imaging, where we can look at skin mechanics in real time, and we can look at vibration, how it propagates through the skin. And we can also look at naturalistic touch, the skin mechanics. And of course, this is also combined with microneurography. And the other focus area is this robot human-like touch. So using a robotic arm and the tracking data from humans, we can start to program the robot to touch in a human-like way. And this gives us a more fine-grained manipulation over the mechanical inputs. Okay, so there's the acknowledgements in the current lab, collaborators within Linkoping University, University of Virginia, and other places. And some of the early work was funded by Facebook, and the current work is funded by Swedish Research Council.
All right, thank you very much, Sarah. We have a second talk that's going to immediately follow Sarah's, but I want to take a couple minutes now. If there are any questions for Sarah that you'd like to ask, we may have a little bit of time later as well, but I think we could field a couple of questions right now.
Could I just pitch in, Greg? It's Francis.
Yes, Francis.
Sarah, one
Yes.
conundrum I always had with what this C-tactile afferent is doing in terms of linking with an affective state around neuronopathy patients. So the only one I've spent some time with is Ian Waterman. And as he's only got CTs and no A betas, he doesn't run around the world in joyous rapture, stroking himself. In fact, you're hard pushed to get any real response out of him with these brushing stimuli and alternative false choices. There seems to be no real immediate percept. So how does that figure with what these nerve fibers are actually doing in terms of any affective state regulation? They don't seem to generate anything.
Well, I mean, my personal opinion is that we don't really know what the CTs are for. We have
That'll do.
Yeah, I mean, the CT effective touch hypothesis, I feel like this, I think it's, it should be called a hypothesis, right? It's sort of the best working hypothesis, best working
absolutely.
explanation we have, right? But I think we need a lot more studies actually to characterize them, their responses to a lot of different stimuli, and potentially using some molecular methods to, target
we need
knock-out or gain of function or loss of function, this kind of thing. I think would help us understand that much better.
Well, they're not like the other two C fibers, the proprioceptor and the nociceptor. When you stimulate those, you get a clear percept of itch or pain. But yours has finally excited a CT, and I don't think anything happened, as far as I recall.
Yeah.
Hi, this is Ellen. I just wondered if given the data that Sarah just showed us, if we should think about this more as the CT system driving valence and the A beta system driving conscious awareness of the social touch.
Yeah, I mean, I think that's a nice idea. And there is a sort of parallel in the visual system, right, as well, that there's sort of two pathways and there's sort of what and where kind of functional division. And I mean, we certainly could have a similar sort of functional division. But I think, again, like relating to Francis's comment, like we still don't really know exactly where that division lies. Yeah.
I'll throw in my pitch for notes and chords. If you stimulate each of these separate afferents, they get a distinctive response to itch, touch, temperature. But the wetness is the best one. If you stimulate a cold neuron and a pressure neuron, you get the perception of wetness. And that perception of wetness doesn't relate to either of the two individual triggers. The Thurnberg illusion is another one where warm and cool give you hot pain. That's what I think we're dealing with here. Centrally, these inputs are integrated. That's where the color comes from. That makes sense.
I guess tying into the CT.
Can I say one thing? Sorry. Well, I think we need to switch to the second speaker. We have an act. We'll have a little bit of time maybe at the end, but I'm a little nervous on time right now because we're a little over. If you have questions, we'll continue after this. Can you guys switch off on your Saad, if you could bring up your slides. Okay, so while Saad's bringing these up, I'll introduce him. Dr. Saad Nagi is an associate professor in neurophysiology at Linkoping University, Sweden. His interests revolve around understanding the peripheral system's role in acute and persistent pain using microneurography, combined with targeted pharmacological and psychophysical approaches. A recent focus area is understanding how the nervous system creates and modulates perceptions linked to the activation of ultra-fast A fibers in health and disease. Thank you very much, Saad, and please take it away.
Yeah, thank you, Greg, and the NeuronS Meta team. I hope I'm sharing the right screen.
Saad looks good. You're on the second slide, I think.
Okay, good.
Great.
All right, so I'll focus more on fast pain. We have some recent data on CLTMRs that I'd like to show. So just very briefly as an overview, the textbook view is that touch is signaled by very fast, thickly myelinated primary afferent fibers, which are conducting in the A beta range, while pain and temperature signaled by slower, thinly myelinated and unmyelinated fibers conducting in the A delta and C fiber range. However, in the animal literature, there is indication of nociceptors in the A beta range of afferents, the prevalence of which varies across species. The definition of a nociceptor is a high threshold sensory receptor, which is capable of encoding noxious stimuli. So when we were setting up a microneurography lab at Linkoping many years ago, we asked a simple question: does human skin have A beta nociceptors? And to do this, we performed electrophysiological recordings from single axons. As Sarah has already explained, I'll just go through this very quickly. This is ultrasound guided. So we visualize the nerve using ultrasound, and then under real-time ultrasound guidance, we insert a very tiny electrode through the skin into the nerve, and we make small adjustments to the electrode. Here's an ultrasound image so from the upper limb. This structure here is the humerus bone of the upper limb. And here you can see our microelectrode approaching this white blob, within which is the radial nerve. So once we've impaled the nerve, we stimulate the vesicular zone with a range of mechanical stimuli, from brushing, indentation, to pinching, to hair tugging, and so on, until we have identified a single axon. And when confirmed, what we see in real time is, as illustrated here, with each vertical line representing an individual action potential from that recorded afferent. Depending on the project and research question, we record from nerves in the upper limb or lower limb, and more recently we have been setting up recordings from the trigeminal nerve as well. In terms of classification, we rely a lot on mechanical stimulation and And for initial classification, a lot of the stimuli are handheld. And once we have identified what unit type it is, we then switch to more well-controlled stimuli. So we start with brushing, for instance, and if a unit does not respond to soft brushing, it will typically have a high threshold to indentation. Those that respond to soft brush will typically have a low threshold to indentation. We then look at their spike morphology and conduction velocity to differentiate between myelinated and unmyelinated types. We also look at their adaptive characteristics using ramp and hold skin indentation and divide them into rapidly, intermediately, or slowly adapting afferents. And within the rapidly adapting afferents, we look at a number of features, but if we had to choose one, it would probably be hair deflection or hair follicle afferents, which responds to remote tapping or pacinians. And for slowly adapting, again, if we had to pick one feature, for us, what works best is to see the regularity of discharge. And then among the high threshold, those that do not respond to soft brush and have high indentation thresholds, we look for subtypes. We identify subtypes based on responses to temperature and chemicals. So typically, in recordings of this sort, they yield receptor types which are either sensitive or insensitive to brushing. Those that are sensitive, like I said, have low thresholds to these indentations, these tiny filaments pressing against the skin where the receptor field is. And those that are insensitive have high thresholds. And what got us quite excited when we were doing this was when we measured their conduction velocities. We found that a subset of these high threshold mechanoreceptors had conduction velocities as fast as the A-beta tactile afferents. We then looked at their indentation coding properties, and we tested a range of forces of indentation, and we found that across a number of different neural metrics, these high threshold A-beta mechanoreceptors were able to encode forces. And since these are recordings from humans, we can ask them how they feel. And psychophysically, our stimuli, the force range we were working in, was perceived as painful. And so when comparing the neural data for these A-beta high threshold mechanoreceptors and psychophysical pain ratings, we found a strong correlation. So we concluded from that human skin has high threshold, very fast conducting primary afferents that encode painful skin indentations. And more recently, with help from Greg's team, we were able to replicate this using a more well-controlled stimuli. It's the Aurora indenter mounted on a XY plotter for better maneuverability. And again, when we tested the force coding properties of A-beta nociceptors, they were able to quite reliably encode into the painful range. Now with these recordings, we can also send electrical pulses through the recording electrode. And when we do that, slowly go up in the intensity of applied current. And we stop when a percept is reported by the participant. We then ask where the percept is, and if we find there is an alignment, a spatial alignment between the physiologically mapped out receptor field and the perceived area of sensation reported by the participant, then it is thought to be a selective activation of that recorded afferent. We then have a questionnaire where we ask the quality and the size of the sensation and so on. And typically for A beta tactile afferents, the percept, depending on the afferent type, ranges from pressure to tapping, flutter, vibration, but it's never painful. Whereas we found that at those same intensities of applied current, around 5-6 microamps, when we activated single A beta nociceptors, generated sharp pinprick pain. Just out of interest, if you do the same for a muscle spindle afferent, there is never a percept. So in addition to mechanical sensitivity, we can also look at thermal sensitivity and look for subtypes within these mechanoreceptors based on whether they respond to temperature or not. And our specific question was whether A beta nociceptors respond to temperature. And we found that a subset of A beta nociceptors respond to cooling. Not all of them, but a subset of them respond to cooling. And those that respond to cooling, they respond not only in the dynamic phase, but also in the sustained phase of cooling. Whereas they don't. So far, we have not recorded from any A beta nociceptor responding to heating, at least within our test range, which is up to 50 degrees. So we then asked whether this cooling response is TRPM8 mediated. And so to do that, we first recorded from an A beta cooling positive nociceptor, confirmed its response to cooling, and then injected high dose menthol around the receptor field of the recorded afferent until the percept of cooling was blocked, at which point we tested whether the unit was still mechanically sensitive, and yes, it was. And then when we tested cooling, there was no neural response to cooling. So we believe these cool positive A beta nociceptors rely on TRPM8 for their cooling response. Thanks to collaboration with Wenxin Lu's lab, we were able to map this to this population, A beta cool positive nociceptors, to a specific transcriptomic class, the HPEP KIT, as the name implies, has a high KIT expression. And in the interest of time, I won't go through all the details. More recently, we have a preprint out showing that this HPEP KIT is a human hair follicle nociceptor. Now I'll talk briefly about CLTMRs and a comment on potential species differences, or whether certain features are human specific. So it's well known in at least animal literature, and there is some evidence in humans as well, that CLTMRs respond to cooling. We were able to show that in our recordings as well. That all CLTMRs respond to cooling. Interestingly, we found that a subset of them also respond to heating. This, to our knowledge, has never been reported in any species investigated. So we think human CLTMRs are polymotor. And we then asked whether the cooling response is tryptamate mediated, and no, it's not. So we think it relies on some unknown channel or different mechanism. We also asked if the heating response is TRPV1 mediated. And indeed, there was prediction from the sequencing data that human CLTMRs express TRPV1. And yes, we found that human CLTMRs, those that respond to heating, also respond to capsaicin, suggesting that probably rely on TRPV1 for their heating response. What's also interesting is when comparing the response of these polymodal CLTMRs with cool responsive nociceptors, that CLTMRs have a preference for temperatures that are innocuous and may be considered pleasant for cooling, as well as for heating. So for both cooling and heating, these polymodal CLTMRs have a preference for innocuous or pleasant temperatures, whereas when you approach noxious temperatures, the response of CLTMRs starts to drop off, and the C nociceptors become more responsive then, as shown here, both for cooling as well as for heating. So from that, we speculate that it's not only pleasant touch. Human CLTMRs may also be involved in signaling pleasant aspects of cooling as well as warming. And just finally, a quick mention of our current focus areas. We are looking at the effect of acute experimental information on primary afferent responses. And we have recently extended this to patients, to individuals with fibromyalgia, recording not just from cutaneous afferents in fibromyalgia, but also from muscle afferents. Planning 2 studies, one involving oxytocin, again, with a peripheral focus, as well as study recording from trigeminal afferents in individuals with migraine. So with that, I'll thank all the people involved in these different studies, and happy to take any questions. Thank you.
All right, thank you very much, Saad. We can open it up to questions now.
This question is more from the data Sarah presented, but for both of you, it just strikes me that all of the emotional hand gestures that were present in your data are neutral or positive in nature. I know one is sad, but I feel like the purpose of connecting while sad is still a kind of pro-social purpose. Like, I think all the even tapping, it varies, but tapping often it's an invitation or it's ambiguous. So I just wonder if you included have considered including a broader range of stimuli. And I was kind of considering, what would that be to be kind of antisocial touch communication? A lot of it would be pain. But those of us with young children probably have some ideas of like communication, negative communication via touch. So just wondering about your thoughts on that, if that might get a little bit more discrimination via CTs.
Hi, can you hear me? I just have to get going to my next appointment, so I switched to
Sure.
mobile. Yeah, but this is yeah. So I think that yeah. To be honest, I think initially that set of 6 was a little bit arbitrary, but certainly I think for microneurography studies, it would have been a challenge to have sort of more negative expressions, because things like you know, anger are certainly legitimately used for communicating with touch. The issue is that, you know, if we introduce too much movement, we could have problems with the recording, where we dislodge the electrode and fail to get the neuron. So I think it would be difficult to capture some of those, studies with some of those expressions with a microneurography study.
Yeah, absolutely. Yeah, I'm just bringing up for the kind of point of view of theory. I'm just thinking about sensations kind of like scratching, pinching, poking that might be, you know, not intensely painful, but more antisocial or irritating.
I mean, there's that yeah, that could be nice to do, actually. I think it's definitely, if you look at the affective touch research, whether it's nerve recordings or something else, there's no there's a lot of focus on the positive, and if we yeah, and I think, yeah, things like pinching and scratching, certainly they can be it wouldn't necessarily be a problem. I mean, Saad is doing this in the nociception experiments, actually. Some of these are unpleasant sensations.
Yeah, thinking of like picking up a little mouse and it's like scrambling and trying to get away or something. But I don't know, maybe there'd be a lot of CT in that too. Thank you.
Okay, others with questions? I'll ask one while people are formulating theirs. There's a lot of people on this call that are dealing with massage, maybe from a clinical standpoint, and might be wondering about a collaborative project. And I was wondering if there's anything that strikes you as off limits from a microneurography standpoint, any kind of deal breakers that you think of right away, that there's no way we could do it like this, and or if there are any resources from a practical standpoint to those sorts of things.
Would you like to take this?
Saad, sorry, Saad, go ahead.
Take it, Saad, that's great. Yeah.
I mean, stimulus control is quite important to be able to sync the stimulus parameters with neural response. I mean, that's that can be quite critical. I mean, these are recordings in awake humans. Stability can be an issue. For instance, the stimulus has to be at a safe distance from the recording needle. If let's say if we're recording from an arm or a leg, and if it moves too much, I could displace the needle, and we'll have to start all over again. Other than that, I think it's the usual ethical considerations of working with a human would apply here as well. I don't know, Sarah, if you want to add to that.
Yeah, I guess just thinking about something like massage, if you're applying some kind of strong force, then I guess just think about how much is going to be moving the limb. You can probably get away with fairly slow movements. And we might end up being a bit restricted to the slow end if you want to apply some high forces. But there are ways also to stabilize the limb, like put in some extra effort to stabilize the limb. I wouldn't say that. Yeah, I don't know what would be off limits. We would just have to try it.
Great, thank you.
How far usually is the needle from the stimulation site, the touch site?
Depends on the target nerve. So for instance, for most of the data Sarah and I showed, the recordings were the recording electrode was inserted just above the elbow, and the stimuli were applied to the dorsum of the hand. So there's good distance between them. Yeah. But it depends, depending on the target nerve. Yeah. For massages, there are, I guess you would want to target the weight-bearing muscles, right? That would be the preference, or maybe the lower
I guess something
more interest, I guess.
Yeah, the more you sort of push up, more probably additional sort of elaborate setup you have to consider. I know that sort of one of our colleagues, Eva Yorotska, has done a bunch of work on proprioception in the lower limb, but and they basically built this whole structure to sort of hold the body in the right way. So it just depends on the time and resources, really.
How interested we are. I thought Francis was going to say something.
How are you getting on with your trigeminal nerve recordings? Because this is where most of these affected C-tactile afferents are housed in the face compared to the forearm, which is not probably the best place to look for them. That psychophysical paper that Greg and I published in the 1990s, just stroking across the forearm and the face, there was a significantly higher recording of pleasure to touch when the stimulus was stroked across the face than the forearm. And in fact, the best place to go would be the back, of course, in ecological. So how are you getting on with the trigeminal stuff? Because that's where this nerve fiber was first discovered, of course.
Yeah, so it's still early days for us. I think for us it's important that we can visualize it reliably on ultrasound.
Right.
I think that that would be great. It's a tiny. The supraorbital nerve is very, very small. We've been putting in a lot of effort. If we can reliably visualize it on ultrasound, then we have a good chance.
Yeah, we did get a unit in my lab in Liverpool in the infraorbital nerve, innervating around where the lip is. But it's all that bone which is a problem with the supraorbital nerve, of course. But,
yeah, it's a problem, but can also be an advantage.
Yeah.
I think that makes the anatomical trajectory more predictable. So.
Thanks.
Okay, I'll ask for a last call for questions before we end. Okay, as a reminder, we will be putting out a call for proposals for collaborative projects in this general area involving microneurography. That'll be tied to the summer school for 2025. So please look out for that. And thank you everyone for coming today. Have a great day.
Thanks, Greg.
Thank you.